morning, dear students. Today we are going to learn about a new topic in the prawn. So that is sense organs of prawn. It is very important topic. Yes, let us recapitulate last class. So, in the last class, we discussed about nervous system organization, the nervous system, central nervous system, peripheral nervous system, and next organization of the central and peripheral nervous system, and autonomous nervous system. Okay, so all these things we learned in neural system of the prawn. The sense organs are part of neural system, components of neural system. So today we are going to discuss about sense organs of prawn. Let us discuss sense organs of. Yes, if we observe the organization of nervous system of prawn, already we discussed in last class this thing. Yes, once again I am going to recapitulate the organization of nervous system. It include central nervous system and peripheral nervous system, autonomous nervous system, and sense organs. Followed. The central nervous system include brain and a double ventral nerve cord and segmental ganglia. Peripheral nervous system include nerves, and the sense organs are compound eyes, and then statocysts and receptors that consent to antenna and antennules. Followed. So today we are going to discuss about sense organs. Okay, clear? Right. Let us see in detail about sense organs of prawn. So, what's the use of sense organs? The sense organs are mainly perceives the stimuli from external environment. They receive the stimuli from environment and send signals to the central nervous system, and then appropriate motor signals sent to organs, affect our organs, then affect our organs to be respond, isn't it? So that is the mechanism involved in neuronal action. So next, the specific sense organs in the prawn are compound eyes, antennules, and antennae. Hello? So remember, these are the main sense organs that executed in the body of prawn. They are compound eyes, antennules, and antenna. The compound eyes serves vision. They are meant for vision. Okay, they are for vision. So the antennules and antenna bears receptors, and they perceives different stimuli. Okay, so different types of stimuli they perceives. So compound eyes for vision. Okay, clear. Now we are going to discuss about compound eyes of prawn in detail. You verify this visual representation. This is prawn, and you have to locate the compound eyes in the prawn. You observe the circle mark. Inside the circle, the black color oval shape, hemisphere shaped structures are seated on stalk. They are called compound eyes. These are compound eyes in the prawn, and you verify the diagrammatic view of the compound eye. So the compound eye seated on two jointed stalk, and next the section view of the compound eye found like this. Okay, clear? So next, you observe the prawn shows one pair black color hemisphere shape compound eyes. They are black color hemisphere shaped compound eyes are present. They seated at the base of the rostrum. You verify arrow mark. The black color stalked eyes or compound eyes are present at the base of rostrum. You verify rostrum, sharp pointed structure, sawtoothed structure. So they seated in the orbital notch that present at the base of rostrum. You verify the clear image of compound eyes on stalks. Followed. Next, compound I is accommodated on short two-jointed flexible movable stalk. So they're seated on stalk. They are two-jointed structure, flexible movable structure. They can rotate in all direction 360 degree. Okay, so this is visual representation of compound I on two-jointed stalk. Followed. Right. The next. The each compound I consist group of conical shape units. They called ommatidia. They are arranged as a radiating fashion in the compound I. Followed. So each compound I, if we verify the compound I, the compound I like higher animals, not a single eye. So the compound eye contain many visual units. The visual units of it called what? Ommatidia. Each compound eye constructed with approximately 2500 ommatidia. So they arranged in the compound eye as a radiating fashion. You verify in the diagram. So this is um, compound I on stalk, and this is section view, longitudinal section view. In the longitudinal section view, you verify. So this is a visual unit, that is ommatidium. So many visual units are there. You verify. So many 2500 visual units are there. They arranged as what fashion? Radiating fashion in the compound I. Followed? Yes, let us verify this visual representation. The compound I consists group of visual units they call ommatidia. You observe this visual representation. So this is compound I, and hexagonal shape facets you verify. They are surfaces of ommatidium. Inside the circle you verify these are visual units. They are ommatidium. Follow? So the compound I consists many visual units. They are ommatidium. You verify in the circle. They are ommatidia. Okay, clear? This is individual ommatidia. Followed? Right. So next you will verify the structure of visual unit, that is ommatidia. It is unit of compound I. So if we observe the structure of ommatidia, the ommatidia include mainly two regions. It is a conical shaped structure, so it include two regions with additional another part. So first region is dioptical region, and second one receptor region, and the third component of it chromatophore cells. The dioptical region include these parts. They are cornea and cornea, and then corneogen cells, crystalline cone and cone cells, and then iris pigment sheath. These are concerned to dioptical region, and the receptor region include these parts. They are rhabdome, and then retinular cells and retinal pigment sheath. And besides these two regions, along the length of the ommatidia, we found specialized cells, pigmented cells. They are chromatophore cells, which forms a sheath, iris pigment sheath and retinal pigment sheath constructed by the chromatophores. Follow? So this is structure of the ommatidia. This flow chart represents structure of ommatidia. You have to write the flow chart in the examination. Yes, let us discuss in detail about the components of ommatidium. So this is compound I, the unit of compound I, ommatidia. They are conical in shape, arranged as radiating fashion. This is microscopic view of the ommatidia, which contain two regions. You observe these are two ommatidia are there in the diagram, right? So it include two regions. The upper side you observe that is dioptical region, and lower side bottom side you observe that is the receptor region. Alright. So the cells of receptor region placed on a membrane that is called basement membrane. You verify the green color arrow mark that represents the basement membrane. Beneath it optical nerve present. The optical nerve innervate across the basement membrane to cells of the ommatidia. Followed. Right, the structure of ommatidia, you observe this is single ommatidia. The parts of ommatidia, let us verify. So this is uppermost layer, that is cornea layer, and beneath it two corneogen cells are present. To verify clearly all the components we are going to introduce here. Next, crystalline cone it is. It is secreted by cone cells, surrounded cells they are, and iris pigment sheath with its dioptical region completed. Next, let us go to receptor region. It includes rhabdome, and next contain retinular cells and surrounded by retinal pigment sheath. All these are seated on basement membrane. Underneath it, optic nerve is present. Followed. These are the parts of ommatidia in detail. Each and every part we will discuss. The first region is dioptical region. The first part of it is cornea. You verify where cornea located. So this is cornea. Verify. Yes, this is cornea. It is a transparent biconvex cuticle layer, and it acts as a lens. It acts as a lens. It is a refractive region. Whatever light rays from the object, they fall on cornea. So it is a transparent layer, acts as a biconvex lens. It is a refractive region of the ommatidium. Followed. So it is derived from ectodermal layer. It frequently undergoes shed off during molting. Right, next, the subsequent part, corneogen cells. One pair corneogen cells are present in ommatidium beneath the cornea. These corneogen cells, also called as lenticular cells. Verify, these are corneogen cells. These are also called lenticular cells. What is the use of corneogen cells? In the name you observe, gen means production. Corneogen cells, they produce cornea. The cornea is not a permanent layer, so during molting it shed off. After shed off, new cornea layer to be secreted by corneogen cells or lenticular cells. They are modified epidermal cells of the body wall. They are two in number, seated underneath the cornea and secretes the cornea. Okay. Next, another component of dioptical region, that is cone cells. You verify beneath the corneogen cells. In longitudinal section view, we found two pillar-like cells, but you verify transverse section. In the transverse section, four cells are there. So these cells also called vitreolar cells. Followed. So beneath the corneogen cells, they are present. These are cone cells, also called vitreolar cells. You verify arrow mark, they are cone cells. You observe green color arrow mark, so that is transverse section view. In the transverse section view, verify four cone cells are present around the ommatidium. In longitudinal section, only two seen. Okay, these are also called vitreolar cell. What is the function of vitreolar cells? These vitreolar cells seated underneath the corneogen cell, and they secrete crystalline cone, a transparent conical shape structure that is called crystalline cone. It's seated at center of the dioptical region, you verify. So that crystalline cone secreted by these vitreolar cells, cone cells. These are cone cells are elongated pillar-like cells. Followed. Right. Let us see the crystalline cone. Right. The crystalline cone, it is a conical shaped transparent part present in the central portion of the dioptical region. It's surrounded by cone cells. You verify this is crystalline cone. Pink color arrow mark, you verify that is crystalline cone. Follow? So yes, you see the crystalline cone. What is the function of crystalline cone? It is a focusing region. Whatever light rays fall on cornea, that light rays enter into dioptical region. Such light rays to be focused into deeper part of the OMMATIDIUM by this unit. So the crystalline cone is focusing unit. You remember? Right. Next, with this we finish dioptical region. Beneath the dioptical region, the receptor region is present. Followed. The receptor region, the first part of receptor region, rhabdome. You observe the central part of the receptor region. So that is rhabdome. Yes, you observe arrow mark, yellow color arrow mark, that is a rhabdome. It is a pillar like structure, transparent pillar like structure located at central portion of receptor region, that surrounded with 7 retinular cells. Around the rhabdome you verify hockey bat like pillar like cells are there. Such cells are called retinular cells. So rhabdome surrounded with 7 retinular cells. Remember, in transfer section you verify 7 retinular cells are conspicuous. That is rhabdome around it 7 retinular cells. It's secreted by retinular cells. The rhabdome lies on basement membrane. Okay, it is a photoreceptor region, and the rhabdomeres of the retinular cells opposes with each other along the axis of the ommatidium and ultimately constructs the rhabdome. Okay, so that is the receptor region. Another component of receptor region, retinular cells. The retinular cells are hockey bat like shape cells or pillar like cells. They are elongated pillar like cells. These are sensory cells. You verify these are retinular cells. They are sensory cells and secretes rhabdome, and 7 retinular cells are present around the rhabdome. They lies on basement membrane and innervated with optical nerve. Remember, so at the base of the retinular cells, you verify optical nerve is there. The branches of optical nerve enter into retinular cells. Followed? So in the ommatidium, the sensory cells are retinular cells. The retinular cells contain specialized units they call rhabdomere units. Such rhabdomere units are forms the rhabdome. Followed? They fuses and forms the rhabdome. The rhabdome and retinulae together forms photoreceptor region. So they send the image of object towards the central nervous system. Right, last and final component of ommatidium, chromatophores. The chromatophores are nothing but they are amoeboid shaped cells, movable cells, pigmented cells. Followed. So they present in dioptical region and as well as receptor region of the ommatidium. So they capable to move from one end to another end, proximal to distal region of the ommatidium. So in dioptical region, they forms iris pigment sheath. You verify. This is iris pigment sheath around the dioptical region. In the same manner, the same chromatophore cells forms one more sheath at receptor region, that is the retinal pigment sheath. So iris pigment sheath around the diaptic region and retinal pigment sheath around the receptor region. Followed. So these two sheaths are constructed by chromatophores. Remember, these are pigmented sheaths. They are useful for absorbs the light rays. Okay, they never permits the entry of light rays from one visual unit to another visual unit, neighbor visual unit. They are protective sheaths. Okay, clear? So this is the function of iris and retinal pigment sheath, right? Next, let us will see the functioning of compound eye. In what way the compound eye functions? The compound eye of prawn or vision organs, they remain in movable jointed stalks and forms 360-degree vision. They capable to pick up motion and peripheral vision. The working of compound eyes are very complex. They have low ability in focusing and clarity of image. So when we compare it with higher animals, like higher animals eye, high definition images won't formed, but the low clarity images are developed by the compound eye. The OMMATIDIUM, the compound eyes are very complex, but they have low ability to focusing phenomena and to establish the clarity image. Follow? So they capable to form motion pictures and peripheral vision. Okay, clear? Right. Let us see the physiology of compound eye. The units of compound eye are OMMATIDIUM. So they are visual units. They forms the images. Such images are uh, observed by the compound eyes, isn't it? The compound eye include about 2500 OMMATIDIUM. The images that produced by the compound eye formed by the functioning of OMMATIDIUM. The OMMATIDIUM of compound eye forms mosaic vision. Remember, it is very, very important. The compound eyes forms mosaic vision. The type of vision is mosaic vision. What is mosaic vision? Let us see for clarification this visual image. And what is mosaic vision? You verify this picture. So you observe object in front, that is object. Actually our eye looks object like this, but behind the object you verify that is mosaic vision. Whatever object is present, so that object appeared as what partial images, as dots, you remember. The hair has so many dots, the face has so many dots, the body blue color, shirt you observe has so many dots. So that is mosaic vision. Mosaic vision is nothing but the complete image is developed by the fusion of so many partial images, like a cinema poster. Right. Larger cinema poster contain two or three or four or six of partial images. Such partial images, when joined, the complete image, complete poster to be produced. So that is mosaic vision. Followed, right? Next, you observe these are all are partial images. That partial images develops full image. Clear? Right. Yes. Let us see the mosaic vision. Each and every OMMATIDIUM of the compound eye does not forms complete image of the object. You remember, in mosaic vision, you remember the OMMATIDIUM never develops full image of the object. Just the OMMATIDIUM forms partial image, small image, part of image of the object. All these small partial images formed by the OMMATIDIUM to be adjoined together and form complete mosaic image in the brain. So, for example, if a prawn observing a object, so the compound eye contain 2500 visual units. So that object developed 2500 partial images. Such 2500 500 partial images joined together and form that complete image of object. So that vision is considered as mosaic vision. I think you followed, right? Let us see the functioning of OMMATIDIUM. The OMMATIDIUM forms mosaic vision. The functioning of OMMATIDIUM is different during daytime and nighttime. So the working of OMMATIDIUM is different during daytime and nighttime. Why? Why? Because during daytime, heavy light, the intensity of light is high. Nighttime, dim light, twilight is there. Low light intensity is there. During this time, the functioning of the OMMATIDIUM is different. You observe this visual representation. So whenever daytime is there, sharp vision by the OMMATIDIUM you observe. So now night time, dim image, conspicuous image. You verify OMMATIDIUM portions, white color and pink color portions. Whence daytime is there, sharp vision is there, and when night time sun goes, and during that time unconspicuous image to be produced. Alright, so let us see in detail about functioning of OMMATIDIUM. Based on light intensity, the OMMATIDIUM of compound eye capable to form two kinds of images due to the movement of the pigment cells, chromatophore cells. I already told the chromatophore cells are move from distal end to proximal end to distal end. They are amoeboid cells, movable cells. Followed. Due to the movement of that chromatophore cells, the OMMATIDIUM capable to form two kinds of visions. So the visions of OMMATIDIUM are apposition image and next superposition image. Remember, it develops two types of images. They are apposition image and superposition image. Now the apposition image. The OMMATIDIUM forms apposition image during daytime. Remember, whenever the light intensity is more, at that time the OMMATIDIUM forms apposition image. So during daytime the intensity of light is more, hence the chromatophore cells of all OMMATIDIUM spread equally around the OMMATIDIUM and forms pigmented sheath between neighbor OMMATIDIUM. They developed conspicuous iris and retinal pigment sheath around the OMMATIDIUM. Okay, clear? So you verify the ash color sheath is this, that is iris pigment sheath, and the next retinal pigment sheath around the OMMATIDIUM. All neighbor ommatidia are completely isolated and fully covered with iris and retinal pigment sheath in the daytime, isn't it? The chromatophore cells or pigment sheath present around the ommatidium never allows the light rays transmission from one ommatidium to neighbor ommatidium during focusing. Whenever from the object the light rays fall on cornea and focused into the deeper part of the ommatidium, at that time the light rays whatever focusing by ommatidium they can passes along that single ommatidium only. They never cross, they never enter into neighbor ommatidium. You verify this visual representation. So these are light rays, followed. So refracted on surface of cornea and focused. So they're moving along the same ommatidia, followed, and not entering neighbor ommatidium. Once again you verify. Yes, that's all. And forms the partial images are fused together in brain and form full image, that is mosaic vision, followed. So the visual units of compound eye can generate many clear definite partial image of an object, and they assemble together in the brain and form conspicuous full image, that is the A position image, followed, right? So this is a position image. It is a clear image, sharp image. Followed? Yes. Next, we are going to discuss another image, that is superposition image. The ommatidium forms superposition image during night time or dim light. During night time, the intensity of light is less, hence the chromatophore cells of all ommatidium migrated at proximal and distal region of the ommatidium due to its some portion of ommatidium, middle portion of ommatidium does not occupied with pigment sheath. You verify this visual representation. This is a pigmented sheath, iris pigment sheath present at proximal region, but you observe middle portion of the ommatidium, there is no covering. Colored. Right. Superposition image, let us see. During night time, the light rays from a visual unit to be passes from one ommatidium to neighbor ommatidium during focusing due to lack of pigmented sheath. So they enter, the light rays are crosses. They enter from one ommatidium to neighbor ommatidium. Let us see the visual representation. So this is refractive phenomena. So the light rays uh, yes, yes. Once again, you see the light rays focusing and crossing with each other, entering from one ommatidium to another ommatidium, by which what happens, overlapping images to be produced. So the receptor region of visual unit receives its own rays and as well as neighbor ommatidium rays. That is the default. You observe the ommatidium, the light rays of own ommatidium not passing. So the ommatidium receiving its own rays and as well as neighbor rays also. Clear? So that's why what type of image to be developed? They generates inconspicuous overlapping partial images of an object, and all these are fused in brain and develops what, inconspicuous overlapping blurred full image. Such kind of image is called superposition image. In the name the super is there, but it is not a clear image, it is a dull image and blurred image. Okay, so that is formed during night time. So with this we completed a position image and superposition image. With this we with this we completed physiology of compound eye and working of ommatidia. I think very clearly uh, understand of this topic. In next session, uh, we are going to discuss about statocyst of prawn. It is another kind of the sense organ and very very important topic, compulsory ask in examination. So the statocyst of prawn image you verify. So in next class we will discuss this topic. Okay.